Okay, so we've been having a bit of trouble circulating the data, and that is largely due because of constraints on GitHub. Some of the files are really big and we just can't upload those. So I do hope that you guys managed to get the files from the drop-down transfer link that we shared earlier this morning. Everybody got that, or should we post it here in the chat for everybody? Okay. So let's get cracking. Okay, so
I just shared the link on the chat, so if you missed it, you can just quickly download.
Thank you.
Yeah.
So in this zip folder, if you've downloaded it, you will see a couple of files and a couple of folders. Let's first just look at the files. So this HTML, if you follow this through to the end, you will basically be able to do the exercise as is. So today we're going to talk about ancestral state reconstruction and how we do import-export analysis. So the crux of the matter is basically we annotate a phylogeny, a dated phylogeny, and then based on sampling locations. And then we start at the root of the tree and we just transverse across the entire tree, and we see or count character states when it changes from one geographic location to another geographic location. And then we also record the time of that event. So that's basically the crux of the matter. And in order to do this, you need a FASTA file with all the sequences, a maximum likelihood tree, and a metadata file containing the dates and the locations in a CSV or a TSV format. And you
Hello, sorry, Eduan.
Yes.
Are you sharing your screen? Because we can't see anything.
Oh, sorry. Okay, can everybody see now?
Yes.
Okay,
yes.
Great.
Thank you.
So this HTML file is also in the Dropbox folder, download folder. So if you click on it, you can just view the content in your web browser. So, okay, so to do the ancestral state reconstruction, like I said, you need these three files, but before we get cracking on that, perhaps let's just look at how we infer a maximum likelihood tree topology. So I prefer to do this in IQ-tree, and the commands are pretty simple. So if you in the folder with the alignment file.
Eduan, sorry, we are still showing the HTML file. I think you need to share the whole desktop.
Okay, so let me stop sharing desktop one. Okay, can everybody see my terminal now?
Yeah, it's good now.
Yes.
Okay, perfect. Sorry about that, guys. So if you're in the folder where your FASTA alignment file is, it's as simple as doing IQ-tree. But first, let's just do IQ-tree.h, and that will give you all the command options for IQ-tree. But basically, it's just IQ-tree-s to specify your alignment file. Then to specify a nucleotide substitution model, and the number of cores you want to use. In this case, because I'm on my laptop, I'm just going to use 8. But if you're using a server, you can use as many as you want. And that's basically it. This will take quite a while because the alignment is more than 20,000 sequences in size. So I'm just going to stop it right here. Luckily, there is a tree file in the folder that I gave you guys. So let's just go back. That's basically how you construct a maximum likelihood tree. So next, we're going to go on to the dating of the maximum likelihood tree, and then doing the ancestral state reconstruction. And this is all done in tree time, which was developed by Richard Nayer's lab. And if you click on this link right here, it will take you to the GitHub page where you can download it. All the installation and instructions are also there. So you can just follow along. They also have pretty good documentation. So if you just click on here, it will take you to the documentation page, and you can then read up on the various tools of tree time. Okay, let's go back to sorry, I lost my HTML. It seems. Okay, so if you're in the command line and you just do treetime.h, you will see all the command line options for tree time. Sorry, I just, okay. Like that. Okay, so yeah, there's a couple of interesting ones. So the .h just gives you the help file. The --tree is to specify the tree file. Dates --dates is to specify the metadata file containing the dates of each of the sequences. Reroute is also quite a popular one, and you can reroute by various methods. The oldest is the one that we're going to use. Clock rate, you can either estimate the molecular clock rate or you can specify a specific clock rate to use. And for us, we're going to use a clock rate of 8 times 10 to the minus 4 substitutions per site per year. And yes, the rest of it is pretty self-explanatory. I just wanted to highlight the most important ones that we're going to be using. Also, sorry that.
So Ed, sorry to interrupt. Is it possible for you to like tell us like how you arrived at that clock rate, like why you chose that one, where to find them if you didn't have for a data set?
Okay, so this is kind of the known rate at which coronaviruses mutate. So you can find this in the literature. Okay. Any other questions? Okay.
There are a couple in the chat if you want to look now or later. I think Daniel is just asking, is any alignment method fine for your alignment? And what is your preference for creating SARS-CoV-2 alignments?
Okay, for alignments, any alignment method should work. I use math, but next align is actually quite good, which is also developed by the next strain group. And with next align, you can basically align hundreds of thousands of COVID genomes on your laptop in a reasonable amount of time. So if your data set is extremely large, I would recommend next align. Otherwise, MAFT is fine. Yes. Okay, so let's just jump back. So the crux of the matter to run this is you're going to need, as I said, 3 files. You're going to need your FASTA file, you're going to need your tree file, and you're going to need your metadata file. So to run tree time, you just do tree time, you specify your alignment, you specify your tree file, specify your, just want to make sure it's dates. Then you specify your clock rate, which is 0.008, and reroute, and we're going to do by oldest. And that should run. This takes quite a long time, so I'm also just going to stop it right here. Basically, the output should look like this. Okay, and then you will see that tree time has identified several sequences that violates the molecular clock assumption that you've put on the analysis. So here you can see that, for instance, this sample from Senegal was isolated early in 2021, but it lacks any diversity. So Tree Time thinks that the sample was actually sampled in 2019 or in the middle of 2019. For other samples, for instance, let's take this Romanian one from 2020. It looks like it was sampled early in January, but Tree Time thinks that the actual sampling date is 2021.3. So in order to proceed with the analysis, you're going to unfortunately have to remove these sequences from your tree. And the easiest way to do that is to do it in R. So I've written a little R script here. And if you just go back to R Studio, you can basically then just read in your tree and your various libraries. Okay. Sorry, this was done on my desktop computer, not my laptop. So Trio is not installed. But basically, if you just read in your tree with the APE package, and then you're going to create a vector with all the sequence IDs of the sequences that you wish to drop from your tree. And then it's just literally one line of code, which is new tree, drop tip, and that's your old tree data frame, the drop tip vector that you just created, and then trim internal equals true, and then you can just write that to file. So if you go back to the folder, you will see that there is a tree called newtree.newig. So unfortunately, you have to kind of do this a couple of times. So I removed those and I reran tree time for the dating, and another sequence from Botswana here was identified as an outlier. So you're going to have to do this 2 or 3 times until tree time is happy. So what tree time does is it outputs several folders with some content. So let's first look at the first attempt. So it creates 2 tree files, a divergence.nexus file and a time tree.nexus. It does a root to tip regression. It calculates the dates of each internal node as well as the dates of the external tips. And then it creates this PDF tree or time tree. And so as you can see here, those outlier sequences are definitely a problem and needs to be removed. Okay, so I've done this a couple of times, and our final tree time run, which I did yesterday, as you can see here, looks quite good. All the sequences in the tree looks fine to me, so I'm quite happy. Any questions up until here before I proceed?
Yes, please. I have a question here.
Yes.
So when you run IQ3 and from the beginning, so you have used already the evolution model with the same time when you run for phylogenetic analysis to get the maximum likelihood tree.
Yes.
So I want here to ask, please, if what's the difference between the Tempest, you know, this software that we use it for the clockliness and for time evolution of sequences, and this procedure? Which is the difference between both methods?
So Tempest is a program that you basically use to see whether there is a molecular clock signal within your data set, i.e. whether or not the sequences do accumulate mutations at a constant rate over time. That's basically what Tempest is for. Tree time, on the other hand, is actually used to transform that maximum likelihood tree into a time-scaled phylogeny where the branches are scaled in calendar time. Does that answer your question?
Yeah, and the skyline plot cannot be done by BEAST from using BEAST Bayesian approach, or it must be only with the maximum likelihood approach?
You can do both. So you can do a skyline plot in BEAST, and we will touch on that a little bit later in the year. Or you can actually use tree time. If you look at the documentation here, there should be, like here, for instance, you can do a skyline plot using tree time as well. Any other questions? No, okay. So if you've done this a couple of times and tree time is happy that there's no more outliers and you've removed them, like I said, tree time will then create a tree called timetree.nexus, which is right here. Okay. So to proceed onwards, you're going to have to convert this nexus tree into a Newick format. And there are a couple of ways to do this. The easiest way is just to open this file in BBEdit or any other text editing software. So as you can see here, this is a Nexus file format. And to change this to a Newick file format, you just delete everything there and everything there. And you can then save this as a Newick file. And then you've basically transformed the Nexus tree to a Newick tree format. Okay, so the next thing would be to actually annotate each and every tip, label, and internal node within this dated phylogeny with some geographic identifier, okay? So to make things a little bit easier, I created a migration.tsv file, which basically just contains, as you can see here, all of the sequence IDs, the region where the sequence is sampled. I also created a new category called neo region, that basically just splits the African sequences into North Africa. That's North America, not. Okay, there's a error. So yeah, basically all the African countries into Southern Africa, North Africa, West Africa, East Africa, or Central Africa. And that's going to be important, as you will see later on. So in order to basically annotate, we just use the migration function of tree time. And this is basically just done this way. I'll just copy it, and it's not going to work 100%. Let's just do that. And then the attribute is basically just the field within your metadata file that you want to assign based on. So in this case, we're going to do by region, and this actually runs very, very quickly. Oh, okay. There is a problem in my metadata file, but let's just say for instance sake that it worked perfectly. It will create a new folder called migration, like it worked yesterday when I ran. And that basically just creates another tree file. Let's first open that in a text editor. And basically, you will see here that every sequence now is annotated by region. So the Wuhan sequence comes from Asia, the Hangzhou sequence also comes from Asia, and so forth and so forth. Okay, any questions up until here? Please don't be shy. Everything's still fine? Okay.
Yes, looks good so far.
Okay. So once
So the new region that you created, is it the one that was assigned?
Yes, Sam, assigned.
So when you ran the migration command, did it take the new region or
no?
The old region?
It went by the regular region. So if we go back to the file here, let's just open that. So there's 5 fields. The first field is the sequence ID. The second field is the region. Then the third field is the new region. So we annotated by region, not new region. Okay.
Okay, so the new region is for later.
That's for later. That's actually for folks to try and redo themselves. So we're just going to do the region today, and I'll show you guys how to do the others, but then folks can do the rest on their own. Okay, so now you've basically got a time tree that is annotated. Yes. Hello, is there another question?
Hello, Edu.
Go ahead.
Yes, so I just wanted to ask something with regards to the metadata. In the current situation, you're using geographical kind of data. What other information could be used for the same purpose? Could one use clinical data to do that?
Yes, yes.
Demographic analysis.
You can use, that's a very good question. You can use any data. So for instance, whether you're working with different hosts. So for instance, humans and bats, or you can use any discrete trait. So for instance, if you're looking within a patient, you can mark samples based on the tissue where it comes from, for instance. This sample may be from the liver, or that sample may be from the kidney, and so forth. So in this case, we're using geographical information, but you can use any discrete data to map to the tree file.
All right, thank you.
Okay, so now we basically have a data tree with annotations at the tips and internal nodes. Now we basically just need to take this tree and start at the root and count each transition where it goes from one attribute to another attribute, and we need to record the date of that. Luckily for us, we've got a Python script to do that. And basically the internal bits of the Python script looks like this. It does rely on Baltic, so that's why in the folder, you will see two Python files that end in .py. The one is Baltic, and you can just leave it right there. The other one is the ancestral changes.py, which is this file that we are looking at right now. Basically, it just reads in your tree file, and you need to make sure that it is called annotated_tree.next. And luckily for us, that is the file name that TreeTime puts out. I'm just going to copy it to that folder right there. Then you also need to specify the most recent sampling date. And in this data set, it was the 20th of April, 2021. And you need to convert that into a decimal. So the 20th of April is the 110th day. And if you divide that by 365, you get 0.30137. Okay, this basically just tells it to count by region and record the date, and then it will output a CSV file with that. So let's try and run that.
Edwin, just a quick question in the chat, whether this can also be applied to partial sequences. Should be fine, right?
To partial?
That is just spike or just another gene.
Yes, I initially created this for work on HIV on the pole region, but you can extend it to anything you're working on. As long as you can create a dated phylogeny, it should be fine. Okay, so just to make sure, we've got our annotated Nexus file there. We've got our two Python scripts there. So basically just to run, you just do Python space ancestral changes.py, and hopefully this should run. And it should normally take about a minute or so. Oh, okay, so it ran perfectly. So it tells you the tree height is about 1 year and 1.8 years, the tree length, and then it also says the number of objects in the tree, meaning the number of internal nodes and external leaves. And transversing across the entire tree, it found 3,044 state changes. And that is basically written to a CSV file. And if you look at this, it will basically just show you the time of the event and the origin and the destination. So if you filter the destination field for Africa, you're basically going to get all the importations into Africa. And if you filter the origin fields for Africa, you're going to get all the exports from Africa to other areas of the world. And so we've done this for region, but you can do this by country, or you can do this by neo-region, as I mentioned, though the metadata seems to be wrong because Algeria is North America and not North Africa. So perhaps just try by country next. So in order to do that, we're going to have to change a couple of things. Anyone want to take a guess what you're going to have to change? Yes? Well, let's backtrack first, okay? Obviously, you're going to have to rerun the migration analysis, right? And you're going to have to annotate by country.
The attribute.
The attribute by country instead of region, yes. And then you're going to have to change this Python script so that it looks for country, not region. And that's basically it. It's as simple as that. Okay, any other questions?
Eduan, question in the chat, what QC cutoff do you use for sequences to include in the analysis?
Okay, so that's why we do the QC analysis before we do the alignment. For our African paper, we only including sequences that has more than 90% genome coverage. We don't include sequences with more than 10 ambiguous sites. And we don't include clustered SNPs as identified by Nextclade. And yeah, I think that's it. Obviously also
And the trimming of the alignment,
yes.
beginning and the end.
If you do your QC at the beginning, obviously check that all the sequences has an exact date of sampling and a geographical location associated with it. Otherwise, there's no need for including it, really. Okay, I think we've touched on that.
Eduan, I also just want to note that maybe if you can open the final file again, I just want to note that in the scripts we viewed last week, we didn't have time to go through this one, but we also have some scripts to visualize this. So participants can feel free to go back to the scripts and try to plug in this file and get nice visualizations on the import-export analysis.
Yes, that's what I was going to do this afternoon. I can't remember where the scripts are now. I think that's on my desktop. Unless you've got it open there in front of you.
Can find it.
But yes, so we didn't get to that part. In the previous webinar, we only annotated the tree, which is basically this dated tree topology that you create here in tree time. Okay. And then there's other small figures associated with figure 2 in the manuscript, and those are the import-export analysis results. So this is a new download. I've did a new download of all the African data last week, and I think we've got now about 11,500 good quality African sequences. And I reran the analysis over the weekend, and so we'll update figure 1 and figure 2 in the manuscript based on these new results.
I can show the script quickly if you want.
Okay, I'll stop sharing.
Can you see it?
Yes, there it is.
Okay, so basically, I usually like to work in Excel because I can control it better, but you can work in CSV as well. This is just a file that Eduan sends to me. And first thing that I do is I convert this event time to a true date in the format that I require, just to make the plotting better, because ggplot will plot by actual date and not decimal date. So this is what this as date does. And I do, so I also in the Excel file, I then annotate a bit how Eduan was explaining. So if you filter by Africa in the origin, you then get the exports from Africa. So I kind of annotate by other regions to other regions, or Africa to other regions. That's your choice, what you want to do. And then I subset this data here, and depending on what I want to plot really. And the cool part about this graph is that we used the alluvial plot. I can't see it anymore. Okay, there you go. So all three of these panels, so panel C, panel B, panel C, and panel D are all from this migration analysis. So if you look at figure 2, you will see those figures in our Africa paper. And in panel D, we use the alluvial plot, which makes this nice kind of stripy, connecting the countries to other countries, or region to other regions. And you can color it by the region that you are interested in. In our case, I colored by Africa to Africa, Africa to other regions, or other regions to Africa. So that's briefly it, but you have the scripts in the links that we shared last week, so you can feel free to go through the data as well. And now you know how to generate the input file.
Okay, thank you, Ria. Any other questions? Everybody got access to the Dropbox download folder. The link is posted in the chat. If you run into any trouble trying to do this, or you want to try and adopt this to your own data, whether it be for SARS or for any other pathogen, please feel free to contact me, and I'll be happy to assist in any way I can. Okay.
Yeah, thank you. And please, if you send us later the video, but because of the last session, we didn't get the video, please. Thank you.
Okay, so Tulio is uploading the videos to YouTube. So we will share each and every video recording with you guys as well.
All the videos are also on the Dropbox. And yeah, the first one is already on YouTube as well.
Any other comments or questions? We've got time today.
Eduan, sorry, you just mentioned that you filtered all sequences with SNP clustered, right?
No, clustered SNP. So those are SNPs that within a small window are clustered close together, and that's normally indicative of sequence quality problems, not necessarily real mutations.
Is there any certain threshold for that, or you just look at the result coming from Nextclade?
Yeah, you just look at the results from Nextclade. Nextclade do allow for some clustered SNPs. As we all know, the the 28881, 28882, 28883, that one codon position. So Nextclade does allow for that SNP cluster. So it has some sensitivity as well to known SNPs or clustered SNPs.
Okay, thank you.
There's a question in the chat, Eduan.
I'll have a look. Could you add a word about the need of inferring the ancestor and what are the limitations in conclusions? Okay, obviously, so there's a couple of limitations. Here we make the assumption that we enforce a strict molecular clock. So we're assuming that all of the sequences along all of the branches follow this mutation rate of 8 to the 10 minus 4, which, as we know, that is biologically very implausible, actually, because the virus does experience different evolutionary pressure in different parts of the world. So that's one limitation. The other limitation is obviously sampling bias. And if you go to the TreeTime webpage, they actually talk at breadth how to deal with sampling bias. Unfortunately, yeah, there's not much we can do regarding the sampling bias. For this tree, we just included all of the African sequences as well as a random background data set of external sequences. So obviously, the results will be sensitive to the sampling bias, but the results that we do get follow what we would expect. So there's at least that. The other option would be to do several trees, and for instance, 100 trees, run the script over all 100 of those and get some measure of confidence for these import-export events. But unfortunately, with SARS-CoV-2, this alignment is so big that computationally that is getting a bit difficult to do. Okay. Any?
Eduan,
Yes.
I just wanted to ask with regards to BEAST. I don't know how we can compare that with TreeTime, and other than TreeTime being easy to run and has got less, less amount of setups to do.
Yeah.
Compared to BEAST. How else would you say TreeTime in terms of performance? How would you compare that to BEAST and your general tech of using TreeTime other than BEAST?
Well, as you can see, TreeTime is pretty easy to use. It is not constrained by so many inputs, as you said. The major drawback is that you will have to, like I said, do 1,000 or 100 of these to resample, whereas BEAST automatically comes with its inbuilt statistical support. So I'd say that's the one drawback of TreeTime, is that it does not have any, or it has limited statistical inference compared to BEAST. Of course, BEAST being computationally far more intensive, so it is a question of what's your data set size and which option or which method is the best to use. Okay, any other questions or comments? I see there's a lot of comments regarding BEAST, and we will be touching on BEAST and the phylogeographies later on in the year. Okay, well, if there's no other questions, I think we can
Thank you.
stop for today. like I said, we will share the video on YouTube with you guys. Okay.